You ever wonder how scientists can look at this gigantic, messy pile of data and somehow pull out one single, meaningful pattern that's been hiding inside? Well, let's get into it, because today we're looking at a really brilliant new statistical tool called OSPCA, and it does exactly that. It helps researchers find the real signal hiding in all the noise.
So to really get our heads around how this works, let's start with a pretty simple question. What makes a great cup of green tea taste so great? I mean, you and I can tell the difference, sure, but how would you actually prove it scientifically? It turns out that's a surprisingly tough nut to crack.
Here's why. A single tea leaf has hundreds of different chemical compounds. So you've got this incredibly complex chemical fingerprint on one hand, and on the other, you've just got a simple ranking from a taste tester. This one's #1, this one's #2, and so on. So how do you connect those two things? How do you figure out which specific chemicals are responsible for that #1 spot?
All right, so here's our game plan for today. We're going to start with the science of taste, then look at why the usual approach gives us a totally jumbled picture. We'll introduce the new tool that separates the signal from the noise and watch it bring order out of chaos. Then we'll see how it can track changes over time, giving us a much clearer view of what's going on.
So, let's jump right back into that green tea mystery. This isn't just a hypothetical, by the way. This is a real case study that researchers used to put their new method to the test.
The setup was pretty straightforward. They took 51 different green tea samples that had already been professionally ranked by taste, from the absolute best at #1 all the way down to the worst at number 51. Then they analyzed the chemical makeup of every single one. Now, the crucial thing to remember here is that the data has an order to it, a clear ranking from best to worst.
Now you'd think you could just plug all this into a computer and poof, get an answer. But when they tried using a standard statistical method, the results were, well, let's just say they were a mess. Okay, so that standard method is called Principal Component Analysis, or just PCA. It's a total workhorse in data science. Think of it like a data summarizer. If your data is a 1,000-page book, PCA tries to find the 2 or 3 main themes that really capture the essence of the whole story. It's a super powerful way to simplify complex stuff. But here's the kicker. When they ran the green tea data through PCA, the result was a complete jumble. The analysis couldn't see a clear pattern at all. It basically said that the best-tasting tea was statistically just like the worst-tasting ones. The standard tool was totally blind to that simple best-to-worst order that we know was in the data. And this is where our new tool comes into the picture. It's a major upgrade, designed specifically for this exact kind of problem. It's called Orthogonal Smoothed Principal Component Analysis, or for short, OSPCA. And this slide really lays out the difference perfectly. The old way, PCA, just gets confused by data that has some kind of built-in order, like a timeline or a taste ranking. And this is huge. It can't tell you if a pattern it finds is statistically real or just random noise. But the new way, OSPCA, has 2 superpowers. First, it's built to understand ordered data, and second, it allows for statistical testing, so you can actually prove that the pattern you found is the real deal.
Okay, so what happens when you unleash this new, superpowered tool on that same jumbled-up green tea data? Well, the difference is just night and day. The result was absolutely stunning. With OSPCA, the hidden pattern just popped out with perfect clarity. The analysis finally showed an order that was, as the researchers said, completely consistent with the original taste ranking. It went from total chaos to a crystal clear picture, just by using the right tool for the job. But it gets even better. OSPCA didn't just find the pattern, it told the scientists exactly what was causing it. It pinpointed precisely 14 statistically significant metabolites, those are the chemical compounds, that were most responsible for the differences in taste.
And these weren't just random variables, they were specific, identifiable chemicals. Things like the sugars raffinose, arabinose, and galactose, which showed up in higher levels in the best-tasting teas, and the amino acid serine, which was found in lower levels. All of a sudden, this abstract idea of good taste becomes a concrete, measurable recipe of specific compounds.
So, we know OSPCA totally solved the tea mystery. But is it just a one-trick pony? To find out, the researchers threw an even more complex problem at it, tracking how living cells change over time.
So for this second test, they used 3 different groups of yeast. The scientists measured their metabolism at 11 different points in time to see how they were processing nutrients. So now the data has 2 layers of order. You've got the progression of time, and you've got the differences between the 3 distinct groups.
And once again, the results were so telling. Standard PCA could only see part of the story. Yeah, I could track the pattern of time passing, but it was completely blind to the differences between the 3 yeast groups. But OSPCA, it was powerful enough to see the whole picture. It successfully pulled apart both signals, the time pattern and the group differences, from the exact same set of data.
Okay, so this tool is obviously really powerful. But let's zoom out for a second, ask the big question. Why does this actually matter for science and, for us?
It matters because science is really all about finding that real signal in a universe of noise. By building in statistical proof, OSPCA makes the whole process more objective, more trustworthy. It gives scientists a tool to confidently stand up and say, hey, this pattern isn't a fluke. It's real, it's significant, and here are the precise things that are driving it.
And the researchers who developed it really said it best in their conclusion. They expect that OSPCA can be usefully applied to any data that has this kind of external information, like a time course or a rank order. Basically, they built a very special key for a very specific but very common type of lock that other tools just couldn't open.
And that just leaves us with this fascinating thought. I mean, so many things in our world have a natural order, right? The stages of a disease, patient recovery rates over time, ecological changes, climate data. What other critical patterns that are right now lost in a jumble of noise are just waiting for the right tool to come along and bring them into perfect focus?